Good evening, everybody. Today, welcome to Life Sciences episode 2. Very short and requested. So, if you have any requests on what you'd like help with.
This is a general call to the house. Please note that the She Needs Power talk is taking place at 7 p.m. in the second quad. I repeat, this is a reminder that the She Needs Power talk is starting at 7 p.m. in the second quad to everyone who wishes to attend. Thank you.
Sorry for that, but if you have any requests on topics you'd like clarity with, please do not hesitate to drop it into the group or basically just our group, and we will get to that. So it was requested that we deal with gene mutations and their effects. So over here, I have a table with base triplets on a DNA strand. So basically, the DNA strand looks like this. Start with T, then A, then C, then T, then C, then T, then C, then C, then A. So we group them into 3, and that's what we call base triplets, which we only find in DNA, right? In mRNA, we call them codons. And then in tRNA, we call them anticodons, only because they are opposite to codons. Right. That was a lot of information. Right. So let's put this in a DNA strand, in a single DNA strand. So basically, we're just gonna get rid of this, whatever was going on there. So we're gonna get rid of that, and so this is our original strand over there, which is what we were given over there. TAC, TCT, CCA, right? So that is what we were given, and that's what we're dealing with at this moment. Now, if you look just above, we have another DNA strand with a similar DNA sequence, but there's a change here. Instead of T, we have put G. Now that is what you call a mutation, right? So that is what you call a mutation, and a mutation is basically, just gonna enlarge this for you guys. So a mutation is basically the change in the DNA base sequence. Can be done through addition, subtraction, or removal, whatever. So it's either you add a G, you add a base, you're removing one, blah 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 blah. So gene mutations can happen in any way. So in this case, we removed T and put G. Not intentionally, but it happens, right? So that's what happened. And that is what we're going to be dealing with today. How does that change affect the proteins formed? Because remember, the whole reason why we have base triplets and are opening up DNA is because of the process we know as protein synthesis, right? So what basically happens there is, you know how your DNA strand unzips, and then I have different ones with bases that are interlocking each other, and then with one DNA, one mRNA comes here, and then it forms its own code based on the code on the DNA strand, right? So it depends on the code on the DNA strand, and that's why the protein formed also depends on the DNA code, and that's why this effect will affect the protein formed. So I just want to remind you guys once again that we move from having a DNA sequence to then having a codon sequence to then having an anticodon sequence, which is just the opposite of that filler there. Then we have amino acids which are fetched by tRNA molecules with certain anticodon sequences. So the anticodon sequence determines the amino acid fetched, and the combination of amino acids determine the protein formed, and that is what we are looking for, right? Now we swipe, and this question is now asking us to write down the base of the anticodon triplet 1, so there's a 1 there, sorry, above. So triplet 1 would be this one over here, TAC. So the anticodon of TAC would depend on its codon. As I said, we move from codon to anticodon. Right. Then that means we have to find the codon of TAC first. And we know that DNA T will match with RNA A, and then A will match with U because there is no T in RNA, and C will match with G. So that is the codon, right? Now to determine the anticodon, that means we have to, again, match that up. So A will match with U, U will match with A, which is that over there, and G will match with C. So that means the anticodon, the answer for this question over here, would be UAC. So that's the anticodon of TAC. Right. Then the next question asks us to give the correct sequence of amino acids for DNA-based triplets 1 to 3. So to get to amino acids, you have to pass through the DNA sequence which they gave you, which is just that or that. We have to get the codon sequence to get the anticodon sequence to get the amino acid sequence. So let's do that. So we have the DNA sequence, check. Codon sequence, let's do that. T goes with A with U, C with G, T with A, C with G, T with A, C with G, C with G, A with U. Because this is what? RNA. Alright? So that's how it works. Then we've moved on to the next stage. Codon's done. Now we have to go and find the anticodons. The anticodon of AUG would be UAC, which is that. And UAC, actually, this question is simple. You don't need to find your anticodons because they've given you amino acid based on your codon. So after you get your codons, then you can get the sequence of the amino acids straight. And you can determine anticodons for later, right? So AUG would be methionine, so that's the first one. Then AGA, which would be that, would be arginine, which is the second one. Then GGU, which would be that, would be glycine, which is the third one, and that is your answer for the second question. So the arrows are to show that we are moving from this to that to that. Very important. So that is just you getting beautiful 3 marks, 2 marks, right? Now we move to the important question of the day. How does that mutation thingy concept that we'll talk about affect the proteins formed? Let's think about this. They say during DNA replication, a mutation occurred in triplet 1, which is TAC over there, and C was now changed to G. And that is the change that I've made there. So, how does this mutation affect the structure of the protein formed? Remember, if there's a change here, there's going to be a change there, and there's going to be a change there. And there's a possibility that there won't be a change here, and there won't be a change here. But if there's a change here, there's a change here, and there's a change here. The change depends on how big it is to affect these two, right? So G becoming G, C becoming G means that this is now C, and now these remain the same, right? So over here now we are going to have AUC, right? And then the amino acid formed is now isoleucine instead of methionine, which is the first one there. So now the sequence changes from methionine arginine to glycine and now becomes isoleucine to arginine to glycine, right? So that's the first thing you're going to mention. Okay, no, that's not the first thing you're going to mention, actually. You have to mention that C becoming G results in the new codon being AUC. So that's where they're going to give you your first beautiful mark. Actually, I'm going to use a different color just to show it's actually a mark. So that's your mark. Then, how does that affect? That means now, also, the amino acid formed is now isoleucine instead of methionine. You have to write that, right? That's your other mark. Now, how does that affect what's next? It affects the sequence in this way. The sequence now becomes isoleucine, arginine, and glycine. So that's another mark. So I'm just adding marks to every point that seems important because I want you to be able to get used to writing every single point, right? So now the sequence changes from that. And the moment the sequence now changes, it means that, what is our final conclusion? Protein structure, or we can say new protein structure, is different due to different amino acid order or combination. And that is where you get your other mark. So it's very important that you understand this topic and are able to break it down so that you can be able to list all the important points when they ask you to substantiate in situations like these. If you do have questions, please don't hesitate to drop them in the group and you'll get helped. See you next time, guys.